What's that about? Anyway, it comes to us from Wai-Hong, the Walter and Eliza Hall Institute, down in Melbourne. That's a Wai-Hong Tham. Wai-Hong actually has a very broad background. Today she's talking specifically about her interest in parasite biology, but she's done very fundamental work in cell biology, protein biology, which currently is very focused around malaria in her work at Wai-Hong, but you can also see a lot of potential interactions here in the ANU community, RSB, John Curtin, welcome, man, and beyond. So the seminar really is to introduce, through an exposition of her current research, the types of topics she's interested in, I think. So she's currently an NHMRC investigator at Wai-Hong, and I'm going to let her tell you about her research interests. Welcome.
Great, thank you. Thanks so much for coming out, and I'd like to just begin by acknowledging the traditional owners on which land we meet here today, and I pay my respects to elders past, present, and emerging. So thank you for coming here today. Today I'm going to do something different. I'm going to actually try to tell you two stories, which I've never done before. So I apologize if I run 2 hours late, but I hope you'll still be in your seats and completely attentive. And just want to say it's really wonderful to see friends both from RSV and also from John Curtin here, both from the infectious diseases, but also the malaria field. And it's really nice to be here today. So I work on malaria, and malaria is an ancient disease, but still, unfortunately, one that causes over 600 deaths annually. And unfortunately, some of the gains that we've made pre-COVID has actually taken a hit as health systems have started to fail within malaria endemic areas. There is a vaccine against malaria, but it has actually very low efficacy, but it is still saving lives. And so there's still really a need to actually think a lot more about what this parasite is doing, think about how we can develop new and better interventions or diagnostics. So the malaria parasite life cycle has two hosts. It has this really giant female Anopheles mosquito that comes and bites you. And then obviously it has the human. And today I'm going to try to tell you two stories across both of these hosts. Within the human, there is the female Anopheles mosquito will inject a form of the parasite called the sporozoite. And the first site of infection is actually within the liver. And within the liver, a single parasite can develop into tens of thousands of parasites called merozoites that then get released into the bloodstream. And really, the red blood cell cycle within the human is what causes all the clinical symptoms. So different human malaria parasites might have a 24-hour cycle, 48-hour cycle, and that would kind of coincide with the kind of cycle of fever and chills as they kind of invade red cells, they rupture the red cells, and all this kind of toxic stuff comes out. And so within the red blood cell cycle, a merozoite will invade and then develop 16 to 32 new parasites, and then just continually do that within the red cell if left untreated. A small population of these parasites then form the sexual stages within the human, what we call gametocytes. And so these gametocytes are then taken up into the mosquito. And what's fascinating is that all of parasite fertilization, malaria parasite fertilization, actually occurs within the mosquito midgut. Somehow they decided that's where partying is going to happen. And so what we're really interested in is how do we stop this cycle? And how do we understand parasite fertilization enough within the mosquito so that we can actually stop the parasites from then moving through all these stages and then getting injected from the female mosquito again. And so I'm going to do two stories today to kind of highlight what they do in both hosts. So the first story is a slightly older story, and it's about a new receptor for parasite invasion. So this is really what underpins how I get up in the morning. I love finding new proteins that really ancient pathogens use to get into our cells as a matter of choice. Then the second story is newer for us. It's a space that we're really starting to want to move into is what about these fertilization complexes? You know, what are fertilization and proteins that actually are involved in parasites? And also then how do you use nanobodies to actually stop that transmission from the mosquito onwards? So the first story is about the blood. And so if you think about Plasmodium, which is a species that causes malaria, there's actually 200 species of Plasmodium. And so it's also a story around choices. So only 6 of these actually have the capability of infecting humans, which are Falciparum, Vivax, Ovale, Malaria, and Nosei. And I work on Falciparum and Plasmodium vivax because Plasmodium falciparum is the most lethal of human malaria parasite. Plasmodium vivax is not as lethal, but is extremely widespread. And as we undergo malaria elimination efforts and you drop the cases of Plasmodium falciparum, Plasmodium vivax cases come up. And so it becomes a real hindrance in terms of full-blown malaria infection. And so in terms of what I wanted to study in vivax is actually the red blood cell invasion. And so the form that we'll mention, there's so many, the malaria parasite actually has many forms. I'm going to throw out terms like merozoite, gametocyte, sporozoite, okinetes. But for the red blood cell cycle, I just need you to remember one term, which is the merozoite, which is this blue little thing here, and it basically invades into red blood cells. And we've always been interested in this cycle is because obviously all the clinical symptoms happen here. So if you can stop basically merozoite entry into red cells, then you can stop that whole cycle of infection. So they're quite small. So in gray, you see the red blood cell, and the blue is the merozoite invading, and then the green is basically signaling, calcium signaling that occurs. After that, it's a very rapid process. You can see that that's like about a minute and a half that happened. And then the red cell had this big deformability that's called echinocytosis, which then basically resolves itself. And this is a cute one. This is taken by the lattice light sheet at Wai-Hong. So 2 merozoites going in there, you know, trying to look cute so that we don't kill them, but they are completely deadly. All right, so this is what, you know, I've done a lot of work on is really trying to figure out how the parasites get in, signaling pathways, receptor stuff as well. But why did I choose vivax? I chose vivax to look at because they have another choice when they go into the human system, is that not only do they go into red cells, they are extremely fussy and they only go into the youngest of your red cells, which are called reticulocytes. So on that level, then they choose through host receptor interactions an ability to then only enter the cells. And for me, this is like a fundamental basic biology question. When we look at the field of invasion for malaria parasites, you can get different views of it. And this is one of my favorite views from the 1970s. And really one of the kind of, really, in my opinion, a higher resolution image of a merozoite contacting the red blood cell. And this is not vivax, and this is not falciparum, but a really chubby zoonotic malaria parasite called P. nosei. And people used it because it was bigger and was easier to image. And so here from the 1978, you can see this round thing here is the merozoite. It has this thing called, labeled R here called the ruptures, and the ruptures carry all these invasion proteins, and it just injects them out right at the apical tip, which is labeled here in A. So you see this little protrusion here, and at the apical tip, it carries the expression and localization of invasion proteins from the malaria parasite to then govern entry into red cells. What you see E here is the surface of the red cell, and you can see, I hope, where the arrow points are basically these really close interactions between the apical tip of the malaria parasite with the surface of the red cell. So it's really these kind of protein-protein interactions that I've been really interested in and trying to uncover how do malaria parasites actually choose which receptor it uses on the surface of the red cell. So if you take it back into a cartoon version, a lot of work has been done with Plasmodium falciparum. And what you have here is on the top part, you see all the protein ligands on the parasite side that have been implicated to be involved in that host pathogen interaction, actually binding red blood cell receptors. And so in Plasmodium falciparum, there's 2 families, the EVAs and the RHs. And you can see at the bottom are the red blood cell receptors that over through the years people have uncovered. So they are in general the glycophorins, both A, B, and C through the binding of sialic acid. There's also complement receptor 1, which is one that I discovered when working with Alan Cowman, when Alex Meyer was maybe not quite in. I think you and I probably didn't overlap as much, but yeah. And then RH5 binding to basogen, and the RH5 interaction is really a really important one in falciparum invasion. If you then look at Plasmodium vivax here, you can see that they have the similar homologous proteins to the EBAs. They've got the DPPs, and then they've got the RBPs, which are basically homologous family to the RHs. But what was fun about it is all these question marks underneath, okay? So where are the receptors and which are the receptors? And so for a very long time in the Vivax field, people work on Duffy antigen, or they call it DARK. And they worked on it based on this observation that when you look at infection across the globe, you have a lot of Plasmodium falciparum present within Africa, but you had no Vivax. And the reason why is that through time, African population are actually Duffy negative. And Duffy negative ended up being used as a P. nosei, a receptor in the initial paper, and then found to then also drive Vivax invasion. So for a long time, this is a very important and critical a receptor on the surface of red cells that actually allow Vivax and nosei to go in. And this is why actually we see no Vivax within Africa, and we see Vivax more in Asia. But I really was interested in this question. I was really interested in the whole, role of question marks that you have. And I think the reason why people don't work on Vivax for a very long time is because we have no long-term in vitro culture. So, you may have heard malaria talks from John Curtin, and also within RSV, you can make knockouts, you can do all these fancy things, but it's all done in Plasmodium falciparum because you can grow it in a petri dish, in human red cells. But for Vivax, we don't have that long-term culture. So the only way we can actually look at the parasite is actually from, field isolates. So it just becomes a little bit trickier. So when I entered this field, I really wanted to kind of approach it in a slightly different way. And so we decided to use structural biology and to pair that together with an ability to develop antibody tools, or basically antibodies that would block invasion that we could then work with field-based partners to then test them in the field isolates. But also at Wai-Hong, we had people who were really interested in serological responses. What are these antigens in Vivax endemic area? What do people actually see in terms of their immune responses, and how is immunity generated there? And so we built this program of over more than 5 years ago to really tackle a basic biology question, but we felt that also that our basic biology program would develop tools that would help with immunoepi studies, and also to provide a whole collection of tools that would block invasion. So the big question that we went for is which red blood cell receptor actually governs the recognition of reticulocytes. So I told you that people were really fond of Duffy antigen, or they're really fond of DARK. But the problem with DARK or Duffy antigen is that when you look at red blood cells of all ages, young red cells, old red cells, DARK is present across the ages. So by definition, it cannot govern that entry into reticulocytes. So we knew really that was something else in addition to DARK that would actually allow parasites, Vivax parasites, to go into reticulocytes. And that's kind of the big question that we went for. And so this was published a few years ago. And so I'm just going to tell you that it's transferrin receptor that we found. And transferrin receptor is present on a lot of cells, but within the red blood cell lineage, it's actually only present on reticulocytes. And a lot of work done, in the early days around exosomes and also selective removal of proteins, from cells was done with transferrin receptor, either in sheep or in rabbits. You know, how do you, as a cell mature, how do you selectively only remove one receptor and not the others? But clearly it was definitely something that was on the youngest of the red cells. And so what I'm going to tell you now is I'm just going to give you snippets across what we found a few years ago and sort of where we took this work in collaboration with a lot of people. So it's not going to be in a lot of detail because it's published, but to just give you a flavor of basically, I was really proud of how far this work went, just beyond looking for a receptor. The transferrin receptor is a human protein, and it's essential because it brings iron into your cells. And so it's a very critical housekeeping gene. So transferrin receptor binds to its own human ligand transferrin. Transferrin is the thing that actually carries the iron in. And so through endocytosis, you see that transferrin receptor will bring in and then release the iron and then gets recycled back up to the surface. But from an infectious point of view, for me, what was really interesting is that it's an entry receptor, what we found entry receptor for Vivax, but also there's this whole body of work that was an entry receptor for New World hemorrhagic arenaviruses. So clearly a receptor that's getting hijacked by pathogens. And I think partly due by the fact that pathogens require iron for good growth. Perhaps that's a reason as well. So in collaboration with a group, Manoj Dur Singh's group, who made this transferrin receptor mutant, you can't actually knock it out, but they were trying to knock it out, and the CRISPR-Cas9 actually introduced a couple point mutations, and one of those point mutations actually just downregulated the expression of transferrin receptor, but we could actually just never get a knockout. And you can see that from that work, you can see that transferrin receptor and its human ligand transferrin is downregulated on the surface. And when they use those cells, they can see that you could get almost the same invasion for Plasmodium falciparum, but you really got a hit in terms of Plasmodium vivax invasion. And so this was a really interesting thing that you could do. And so these are actually Vivax isolates taken from Goa, but then the entry cells are basically these knockdown cells. But for us, what we wanted to do was to try to get an insight into what this would look like structurally as a way of trying to understand where we can make antibodies against the parasite ligand. So in this case, then we moved into cryo-EM. And so what I'm going to show you next is basically a 3-component complex. It'll have transferrin receptor with its human ligand transferrin and the parasite ligand. And so what we have here is transferrin receptor is in green, and it's actually transferrin receptor dimerizes. And sometimes if you just think about it in the other images, it'll just look like a receptor. So it has like the apical domain here, the dimerization domain. And then in teal, what we have is basically transferrin bound to transferrin receptor. And what the parasite ligand has done is to kind of hug the side of the transferrin receptor transferrin complex on the side. To orientate you, I don't have the membrane, but the membrane would be down here. So transferrin receptor obviously has a transmembrane domain. And so does the parasite ligand, but these are all extracellular proteins, but the membrane would be down here. With cryo-EM, we could only get visibility over 1/3 of the parasite ligand. There's still 2/3 of the parasite ligand that to today, we still haven't been able to actually visually image using any kind of structural methods. But what we're really pleased about is that we feel that a lot of the receptor engagement is actually being governed by these interactions that we see with 1/3 of the protein. So what you can see here as you rotate through is that you get a lot of the extensive interactions are actually at the transferrin receptor site. And then there's also a binding site here for transferrin as well. So it's actually engaging both of those proteins. What's interesting is that, so it's got a couple of human ligands, right? So it's got transferrin, I talked about that. If you've got iron overload disorder, then this is how hemochromatosis factor binds to it. And then more recently, heavy chain ferritin. But this is where infectious, viruses bind. So basically, RBP2B binds both at the apical domain and also this binding site with transferrin. But Machupo GP1 and some other neural hemorrhagic viruses, they actually bind to this apical domain. And the reason that's really fascinating, we've done a lot of mutagenesis here, is that it binds here because if you look to see where transferrin is, you can see that this domain is completely unbound by the human ligand, except for ferritin. And so we think that that infectious diseases or pathogens have actually kind of hijacked this domain here as a way of entering cells that actually have ways of getting iron into the cells. And I won't have time to tell you this, but there's a lot of structure function analysis that we've done with transferrin receptor, and it actually has some critical residues that is shared with neural hemorrhagic arenaviruses as well in terms of engagement. So if you take the structure apart, and what this drumstick is I've just kind of taken that purple bit and pulled it apart from transferrin receptor and transferrin, and you can see that in green here is just basically the surface area that engages with transferrin receptor. So you can see that most of it is actually governed by that transferrin receptor binding site. And then in blue here, these are the residues that actually bind to transferrin as well. So it's quite interesting that, that most of it is bound here. If you look at the homologous protein, this is actually the proteins that drives homology across all the binding red blood cell families. But this is the first time that in this family of proteins, this part of the protein is being associated with receptor engagement. So Vivax is actually super polymorphic. And so if you look at then genomes, parasite genomic sequence from Thailand, Papua New Guinea, Cambodia, and Brazil as well, then unfortunately, the part that's really polymorphic is actually the part that actually binds receptor, and is actually also under selection. And so this, in some sense makes sense, but obviously then becomes an issue in terms of how we think about our antibodies as well. But what was really interesting is that because obviously we've got the structure from cryo-EM, you can start to map these polymorphisms on here. And this is just showing you one side. But we've done a lot of structure function analysis where the polymorphisms in pink actually don't overlap with the binding site. And we've done the reciprocals as well. And so this actually is a very polymorphic region under selection, but doesn't seem to be hitting a lot of the receptor engagement sites. And so this is something that, so we made RBP, so a lot of my work is structure-based, but part of it is also just kind of generating the antibodies and then seeing if they can block invasion. And because it's really hard actually to do these invasion assays with live parasites, we did have a whole bunch of in vitro assays to test what these antibodies do, that they block red blood cell binding, they block receptor engagement using a FRET assay, and so forth. And then what we did was to then work with our collaborators in Thailand and in Brazil to actually look at live isolates. So when people do Plasmodium falciparum work, because we have long-term in vitro culture, whenever we test these antibodies, we tend to only test them in a lab adapted culture that's been here for a long time. But in Vivax, every clinical isolate is different because everything that you do literally is a separate clinical isolate. And so here, what was really interesting, although, if you're used to Plasmodium falciparum, these graphs don't look that exciting because they would just, be at zero. But the fact that each one of this was a clinical isolate showed that actually our RBP2B antibodies could actually block Vivax invasion. And that's the antibody isotype, and that's the pre-bleed as well. So this was really a good foundation to say that actually these antibodies do work, even in an area that has a lot of polymorphisms within Vivax, and at least we're being able to generate some antibodies that actually can block invasion. The other thing that we did was that we also went in and looked at across different transmission settings. So in Papua New Guinea is a very high transmission Vivax setting. in Brazil as well, but then Thailand less so, but also across young children and then also cohorts of adults. And what was interesting is that RBP2B antibodies are correlated with clinical protection against Vivax. And I think for a very long time, people in the field just didn't make the recombinant proteins. They didn't try to look at, a different, they did, but in terms of invasion, they were very focused on dark and Duffy binding proteins. A long time ago, Mary Galinsky did look at the RBPs, but not in a very, kind of comprehensive way. And we really wanted to bring this family in to really try to understand, what the serological profile is. And so, and we also did a lot of work of using single B-cell cloning to pull out human antibodies from people who have a natural Vivax infection, and to use RBP2B to pull out these human antibodies and then to understand whether or not they could block receptor engagement. And then basically they do, and I won't have time to show you this, but a lot of structural biology went into understanding where these human antibodies bound and which one could actually block invasion. So I think there's definitely, I think RBP2B is definitely a target of natural immunity, and people who are infected with Vivax can actually mount an immune response and generate antibodies that can block Vivax infection. But what's really been interesting about this work is then by just, kind of making the recombinant protein, and wasn't just RBP2B, we made the whole family of RBPs, that then our collaborators, Ivo Muller and Rea Longi, they are really excited about building diagnostics for Vivax, because Vivax infection is very different from falciparum infection. So if you were to diagnose falciparum, you take a blood, you smear it, and then you can see it, or you do, there's enough of a parasite load. Vivax is a very interesting biology. It has a latent phase in the liver. Falciparum doesn't have that. So you can actually get relapses. Studies done in Papua New Guinea show that 90% of infection that they see for Vivax are caused by relapses. So how do you then, how do you actually go into a field site? Their idea is, how do you decide who to treat for Vivax infection when you can't actually detect parasitemia, but it's actually there? And so Rhea and Ivo Muller have been really interested in developing a diagnostic test. And so what they found in the Nature Med paper that they published is that RBP2B, which literally I think it was because we were making the protein next door at Wai-Hong, they're like, yeah, let's definitely run this as part of the panel. And this ends up being one of the biggest targets where you can see a real difference between no detected infection and then someone infected within 9 months. So they collaborated with Michael White and did a whole bunch of modeling and trying to understand how many antigens do you need in combination to actually, decide on prior infection, and how can you predict that someone's actually going to potentially relapse, because the parasite goes and hides out in the liver. And so the person is carrying latent parasites, but you can't test it, you can't see it. And so how do you detect within 9 months that someone's actually going to relapse, and would you treat or would you not treat? And so they've got modeling attached to, different treatment regimes and so forth. So I'm really sort of in a way really proud of this work, because we've really tried to bridge just, that very standard, understanding host pathogen interaction, doing the cryo-EM, to actually people actually going out in the field, understanding immunological responses, and then potentially taking this into a panel diagnostics. But what I'm interested in that we are still trying to work with is basically why reticulocytes? Why do parasites choose reticulocytes? They're only 1% of the circulating blood within the bloodstream. So it's not really a very, it's not the majority of red cells that circulate. But I do think that they're really interesting things. They're super flexible. So are they taking some remodeling purposes? There are some papers out there around exosome signaling. A lot of work now that people are doing are bone marrow reservoirs, is how do parasites get in and access these really young cells that are present in the bone marrow, and what are they doing in terms of regulating immunity. And then obviously, because they're still more metabolically active than mature red cells, how do they perturb then drug-resistant mechanisms that are present in reticulocytes? Okay, so that was the red blood cell story. And then now I'm gonna tell you about the guts part, which is the mosquito midgut. And the reason why I'm interested in this is because, as I mentioned before, all parasite fertilization happens in the midgut. And so parasite fertilization happens, and then the parasite then decides to make a whole bunch of different forms. Obviously, after fertilization, you get the zygote, they make the okinete, oocyst, the oocyst rupture, and then they, in the salivary glands of the mosquito, then you get the sporozoites getting released and biting another human. So if you can block fertilization, then you can actually block that transmission, basically going from mosquito to parasite. And what's interesting about the stages is that it creates a bottleneck in terms of parasite numbers. So I talked about the red blood cell stages, and you can see here from parasite numbers, red blood cell stages are really hard to target because you can get this, ability to just make a lot of parasites in the red blood cells stage. But as you go into the mosquito stages, you can see that the number of parasites that are present actually within the mosquito is actually quite low. So this becomes an advantage in terms of trying to target parasites within the mosquito stages. So we have these things called transmission blocking vaccines. And so basically they give you a vaccine against a fertilization antigen or something that's involved in transmission. The humans make the antibodies, the mosquito bites the human, takes up the antibodies, and then parasite fertilization or transmission is stopped within the mosquito. So this was something that is actually in active work right now. But if you look at the transmission blocking vaccines, there's really around 4 major ones that people sort of work on, 3 major antigens and then a Vivax antigen here as well. They're kind of intriguing. The transmission blocking antigens are definitely less polymorphic. As I mentioned before, you have a lower parasite load in the mosquito, so that's attractive. And also you might prevent the spread of drug resistance mutants because you're not in the human, you're kind of targeting the mosquito stages. So, some really nice advantages there. More recently, they also did like a monoclonal antibody to try to deliver them within humans so that they can block transmission seasonally as well. And so, that sort of work is happening. Okay, so in the mosquito midgut, this is kind of what's happening here. So you get the fertilization, you get the gametes, and then the zygote, and then you get then the okinete here, and then you get the embedded oocyst. All right, so like just also kind of fascinating, all the different stages, all the way down to the sporozoites, which then migrate to the salivary gland of the mosquito to then get injected. I'm really interested in these stages here. How do I stop basically gamete fusion? How do I stop parasite fertilization? So in terms of fertilization, there are a few parasite ligands that people know of already. I guess for the plant biologists here, HAP2 is really interesting. HAP2 was found, I think, initially in lilies and then also in Arabidopsis, but it's a gamete fusion protein that we also use in malaria parasites as well. And then you have the post-fertilization antigens, but it's really a handful of antigens that people have talked about so far. So today I'm just going to focus, we are starting, one of our main things that we want to do is uncover new proteins, but today I'm just going to focus on a really old one that the field has used, PFS 230, because we needed to develop the tools for this. And so I'm just going to introduce this story as a way of trying to talk to you about literally just feeding alpaca antibodies to mosquito is like my main reason for showing this one. But as I mentioned, PFS 230 is like old. People have known about this antigen for a really long time. Big protein. All these boxes here are just the structural domain called 6 cysteine proteins. We have in our lab a real interest in 6-cysteine proteins for a variety of reasons that I won't talk about today. But even after all this time, people worked on domain 1, and the squiggly line is what they call the pro-domain, and they worked on this domain for like a very, very long time. It's, definitely, a target of human immunity to block transmission, and there's a whole bunch of papers using polyclonal antibodies and some monoclonal antibodies to show that PF230 blocks basically transmission. So what we wanted to do is that no one's done nanobody work in this space. And what we want to do is to take nanobodies, which are a special kind of camelid antibodies, as a premise to start to target all the transmission blocking antigens. And hopefully I'll show you why nanobodies are really kind of special to me. And I know very special to Joe Brock's group as well. But they're really small. And the idea is as we start to uncover neutralization antigens or old ones, we can link them together. And so we start to develop antibody therapies that don't just target one antigen, but multiple antigens. And can we do that? We actually also published a new structure of PFS230 that has 2 of the domains. And that is actually a new crystal structure within the field. So, but we move on to nanobodies. And so nanobodies were found many decades ago in Belgium. They are something that occurs in the camelid family, but also the woebegone shark. So when you think about human antibodies have heavy and light chain. Heavy and light chains bind to your antigen, okay? That's how antibodies recognize the proteins. But camelids and also the woebegone shark, they only make the heavy chain domain, okay? So this is very fascinating as an antibody entity because it's very small. So it's very small, as I mentioned, but also because it doesn't have to bind to another partner, it has actually mutated certain things in the scaffold to make it extremely stable as well as an entity itself, okay? And also rather than worrying about the design of how these 2 heavy and light chain binds to antigen, we only worry about one module. So, everything design-wise becomes kind of interesting. Very stable, very stable across pH and temperature range. So then can we then think about different administrative routes in terms of antibody therapeutics as well? The other thing is that because it has actually evolves differently, it has this longer CDR loop. And so this is the human heavy chain domain, and this is the nanobody domain, which is shown here in black. And it's one of the antigen binding loops called CDR3. And it is known that nanobodies can accommodate a much longer CDR3 loop. And why would that matter to you? It matters because it can then access different kind of architectural space within your protein, can maybe go into grooves a little bit better because of that longer loop. And so it's, all of these things become really fascinating in terms of nanobodies. And it is used in clinic for blood clotting disorder. These are 2 nanobodies here joined here by a linker. And then in February 2022, the first CAR T-cell product was also approved that was a single domain CAR T-cell design as well. So definitely a very interesting antibody entity. I personally think will be used more and more in the clinic. So at Wai-Hong, I established like an immunized nanobody platform in 2019 because we know that the yeast synthetic library was being used, definitely within Victoria, but also at ANU. Obviously, you have access to that. And we thought it would be worthwhile to bring in an immunization platform so you can harness the immune system of the alpaca to generate, antibodies that are specific to your target. So it's a very old style platform in a way that it has, a 45-day immunization. After the immunization, we get the alpaca blood from the vet, and then we clone out all the nanobody genes associated with that immunization. And then we do 2 rounds of phage display, and then sequencing, and then nanobody purification as well. So with PFS230, we immunized with recombinant PFS230, and we got 13 unique clonal groups of 13 nanobodies. really high affinities, picomole to low nanomolar, nanomolar. And basically through epitope binning, we found that these two nanobodies kind of basically bound in two different classes across PFS 230. But we just wanted to basically take the nanobodies into the insectary. So at Wai-Hong, we have an insectary, and what's really nice about an insectary is that we can actually breed an Anopheles stevensii. And Anopheles stevensii is one of the mosquito vectors that would actually take Plasmodium falciparum. And so here you have in the insectary a mosquito cage. That's just a sugar wick. And you can see that we have the female mosquitoes are sucking on some blood up here. And then you can pipe in warm water, so it's nice and cozy like a human. And then that warm blood, they just really attracted to the blood. They take up the blood feed. And obviously, if you had infected parasites here, that's what they're taking up as well. And then they go crazy here because Haley's kind of breathe on them, put all this carbon dioxide, and then they just gone nuts in response to that. But yeah, so basically, these are the gametocytes. These are the sexual forms of the malaria parasite. And what we want to do is feed them to these mosquitoes and then see if we then generate infected mosquitoes and can we stop that? So that's a transmission assay. So basically, we take the gametocytes, okay, we feed them to the mosquito, and then after a week, we stop our experiment here by counting the oocysts. So the oocysts is the form of the malaria parasites that sits on the mosquito midgut. So for us, obviously, we want to feed them blood, but we want to feed them these alpaca antibodies and then the sexual state parasites, okay? So hopefully the parasites get incubated with the nanobodies, gets all taken up into the mosquito midgut. Some really exciting chemical cues and temperature cues, pH cues happen in the mosquito midgut, causes the sexual forms to come out. We're hoping that if they encounter a nanobody, my nanobody is just going to stop all their fun in the gut. Okay, but that's the plan. And then this is what you count. You take out the mosquito midgut, and then you can see this hairy thing here is uninfected mosquito midgut. And you know it's infected because it's got these red dots here, and that's the oocysts. Okay, so that's what we're counting. And so we're just trying to see, like, can we, at this stage, hoping then we block these other stages as well. So we did that. We took just, these are wild type oocysts, and mosquito experiments are messy. You get a range of like infection. Ian's graphs might be better than ours, but this is what we get. You just get some range of infections. Sometimes you get no infection, but you get a range up to 40. PBS sometimes does affect transmission, so we always do a buffer control for where our nanobodies are. WNB7 is a nanobody FC fusion from some of our COVID work that we were doing. And so you can see that they don't block. This is basically wild type control antibody buffer control. And then here, LM4 is a classic PFS 230 human antibody control published from a different group. And then these are our nanobody FCs. So really exciting, our first kind of transmission blocking experiment. After coming out of the dark ages of the red blood cell cycle, we were so chuffed to see this as well. And then, so that was really fun to see that you could block transmission. But then there's this other thing that malaria parasites do that kind of cute, and it's called exfagulation. And it's this crazy thing that the male gametes kind of do. And it's historically kind of fascinating because Laverian, who noticed them when he took a blood draw, he saw these exfagulating parasites, which like, it's so hard to see them, but somehow he saw them. And then he decided that actually it's an epicomplexan. But basically you can think of them as 8 male sperms. They kind of bind to this ball. And then under chemical cue and temperature change and something like that, they then get released from the red blood cells. And when they get released, they do this like, kind of craziness, like, whipping around and try to pull in red cells around them. And classically in this field, you can also start to see if your antibodies can block them interacting with the red cells around them, okay? Somehow, I don't know, a male mating behavior, whatever. But yeah, okay, so this is lattice light sheet. Here is a male. okay, stuck in the red cell, and then all of a sudden, boom, comes out. And that's what it does. It kind of goes out and tries to get all these red cells in there, and that's called exfagulation. So what you can do then is you can also then put the nanobodies in this in vitro assay and then look to see if you can stop exfagulation as well. And that's what we did. And we found that together with LM4, our nanobody FCs could also block exfagulation. And this is just trying to, figure out the different stages of mating that, or basically parasite fertilization as we go through these assays. Because we're structural biologists, I have to show you the crystals. And so obviously every nanobody we always do a crystal structure. And so I haven't shown you a lot of crystal structures, but I want to show you this one. So basically PFS 230 binds to domain 1, 2 different nanobodies, both of them binding to different sites of 230. So really interesting that basically the nanobodies can bind these 2 sites as well. What was a little bit tragic was that they also bind the same site as human antibodies. So it didn't look like they were accessing something different. The difference is that they can bind, they can't bind the presence of the prodomain, and that's the mechanism that's slightly different from the human antibodies. But unfortunately, the human antibodies kind of beat the nanobodies in this case. But we'll move forward and be better. Yeah. But really, what's fun about the nanos? I think what's fun about them is that you can obviously this is human antibody. We do put an FC on, so it is Craig.
Yeah.
Seriously, yeah. This is director's lecture.
I know.
Yeah.
Thank you, Wai-Hong.
Yeah, you're welcome. Yeah. So basically, a nanobody can put to the FC, which we have done. So this is not as small, but you don't have to worry about the light chain. But nanos, lots of fun things, as I'm sure you know from a lot of work done here, and you can link them in different ways, And so with our SARS-CoV-2 work, we make these things called bioparatopics, which are, 2 different nanobodies binding to 2 different parts of spike, and that can actually give you broad spectrum neutralization. So, there's lots of things you can do in terms of the modular design of this. And I will stop here. I hope I've given you a flavor of the way I've approached the work, but really where we're interested is to really look at core fertilization processes and how does gamete fusion occur, which I think has a lot of links with, plant biology. And also I think the fertilization complex is going to have so many links with membrane protein biology that's done a lot here at ANU. And here's just to say thank you to everyone. And it's really nice to give directors seminar here, and I take your questions. Yeah.
It's just fascinating. So we'll go straight to questions from the floor up the back.
Nice talk, Wai-Hong. I just want to go back to the transference stuff. Obviously, not as interesting as you question. With the transference stuff, so you say it's transferring 1 receptor. So, yeah, so and that's only expressing the particular sites. So the remain salters and then mature advice or not.
Yeah, so is that asking about transferrin receptor and whether it's only on the surface of reticulocytes and what happens in mature red cells. So as red cells mature, transferrin receptor is a receptor that it terps out. It selectively gets rid of. And so when you look at, so it'll be a spectrum. So even when you purify reticulocytes using Percol, you will always get some mature red cells, and what you see is that you get really high levels of transferrin receptor on reticulocytes. And when things are really mature, then there's no transferrin surface expression. Yeah.
So do you think, because I know you put that diagram where you showed those receptors are not known, do you think that Vivax is what we use is a lot of the passage and as well? So just extend
I think that the Vivax people, there's some really fascinating Vivax biology, and I think they really were so just like one receptor. It's not true. I think Vivax is as promiscuous as falciparum. And I think through time, so since that paper, CD98 is another receptor. It's not reticulocyte restricted, but it's another receptor that they found for Vivax. Basogen, they have hit it with anti-Basogen antibodies, and they don't see such a inhibition with Vivax. But Will there be other question marks to be filled? Yeah, I think so, sure.
Gil.
Yes, that was really great to point out. So I'm curious about the, so I guess you show that the transferrin receptor is essential, and then other work has shown the Duffy receptor is also essential, Duffy antigen is essential. Do you think they need them both? In which case the parasite needs more than one receptor in order to gain entry? And how do you see the interplay between them?
For sure, Duffy is critical. Like Duffy is critical definitely in like a mechanism of maybe getting in. You know, and but I think what it needs is, so I see a couple of things. I see choices in the cell. So I think for sure transferrin receptor will give the choice into retics. And then you need these conserved mechanisms of just getting into membranes or fusing or whatever, you know, in terms of parasite entry. And I think Duffy is involved in that kind of very universal mechanistic thing. Yeah.
Yes.
I guess lots of immune cells have CD71, but activated B cells have lots of CD71 on the surface. They also have lots of dark. So why is Vivax not running around invading activated B cells?
So Ian's asking this thing. I tried to tell you the story about transferrin receptor being restricted, and but transferrin receptors, you know, it's a critical protein, so it's on a lot of cells. And I think comes back to Gil's question about like, you know, what are the co-receptors or what are the other things that Vivax needs? So I think it's a combination of proteins that actually then work together on the surface of the red cell or the reticulocytes that then might also then restrict them into the entry into, let's say, why is it not immune cells, things like that.
Yes, to follow up, I mean, like for super again, like very much more highly expressed on red cells and the cell, but Vivax is going into the immune cells, not necessarily not present in red cells.
Transferrin receptor.
I didn't have transferrin receptor.
Yeah.
I mean, but relative to like,
yeah.
So
yeah.
It's
definitely different. And there's always this question is like people are like, oh, more receptor, better entry. But in the end, you've got only an apical tip. You've got one marrow, one merozoite. And, you know, if you think about protein-protein interactions, there's really, you know, one protein and one person, I guess. But it's the same question around Basigen as well, right? Basigen is actually present on a lot of cells. You know, so how does falciparum decide only the red cell using Basigen only essentially on that red cell? So I think a lot of unanswered questions about co-proteins that then govern just entry into red cells. But your point is valid, yeah.
Other questions, right up the back. Mark.
in the studio, you've got and it was found that kite is parasites mice. And they showed that when you gave lightning with a L7, which is a part of the physical, that was what you with your antibodies. I mean, this is a lot of work, but I think
I haven't heard. So it's asked about chitin and chitinosis, and I haven't heard. Yes.
I do have one question online here. Thanks for that. Is there any possibility to target HTR ligand as a vaccine candidate?
What's HTR? Any of the malaria people know?
Can anyone? I'm afraid I don't know.
Okay, HTRF. Yeah, we try not to do that because obviously based on the fact that it's on 200 cells. So we normally don't, although with the COVID work, some people are really trying to target ACE2, but for our malaria work, we've always try to really not mess with the human receptor. Yeah.
Okay, we have time for one more question. There you go.
I think you showed a structure earlier on. I think it was RBP2B hugging the receptor, and you said that maybe 2/3 of the structure dissolved. Do you know much about the function of that, the rest that you can resolve? Is it enough just to the structural bit that you showed binding on, is that enough to inhibit?
Yeah, and so I think there's some things that we notice. We know that we've made antibodies across all of the protein, and we get these really weird phenotypes with antibodies, that there are some antibodies that enhance binding. They don't necessarily, and for 2B, we never did that work because, you know, it's too hard, but just using recombinant protein binding to reticulocytes, you know, or like a FRET assay, you know, to look at protein-protein interactions, we see that some antibodies actually see an enhancement, and we don't understand that. All of these ligands, apart from RH5, so the homologous family proteins are RHs and RBPs, like RBPs in the viaducts here, they're these big proteins, you know, and I sort of feel like they must do something, you know, maybe it is actually, you know, when the membranes come together or something, and that's what they're doing. So I think it'll be really still fascinating to look at them, because apart from RH5, all of them are really quite big. They're over 200 kilodaltons. So what's the rest of the protein doing? It's kind of interesting. Yeah.
I can follow up with one last question. So one of the things you showed with the work on the sequence variation of the protein and the signatures of selection is there's a lot of selection going on, enhancing diversity, but not in the sites that are involved in the binding. So do you have any hypotheses on what might be driving that selection biologically?
Yeah, I think always a tricky question. I think, like, I was a bit surprised, you know, I really thought when we saw that selection, you know, it would be within the binding site, and I'm not sure. And you know, the protein-protein interactions, like, they're transient, right? They're not, it's not like it gets glued on and the immune system can't see it, because proteins come on and off, and things like that. And so it was interesting to us. We felt like that was good. It didn't do that, but it's not something we expected. Yeah. Because, yeah.
Well, thank you. So please join me in thanking Wai-Hong once again. We'd like a bit of time now, sort of half an hour or so, just for casual discussions with Wai-Hong, if anyone would like to take advantage of that. Perhaps just let her pack up here and then chat with her outside in the evening. So thank you all for coming. Terrific start to the year. I hope to see some of you on Thursday. Excellent. That was